today, and I think part of the question are really bridging to some of the discussion I will have today, is really to try to understand this dynamics, this complexity of cells growing into tissue and organs. So I think what is our ultimate goal for cell and developmental biologists is to understand decision-making, both in development, during regeneration, and disease. And we heard in the previous talk there are a lot of single-cell technology, especially genomic technologies, that now allow us to build these pseudo-trajectories, or in pseudo-time, that really bring and map the development of some tissues. However, this is still not a single cell view, because if you really look at it, you have a single cell is still, we don't know what's the dynamics of cells moving through this trajectory and how really they go from one state to the other. And especially in this decision moment, these are very fast and transitory programs that are run for a very short amount of time. So are we capturing all of these states during the development, and can we really find ways to just have this really mapped well? Moreover, in this bifurcation during trajectory really have a lot of variability. Some of it is still technical because we cannot really map every single cell, but also is really biological. In every decision-making transition point, there is more variability between cells. And how can we map this, understand the sources of this variability, and what are really the consequences of it? So one aspect why it's really important is because cells and our tissue are not built like robot or machine. They're not built top-down. It's not a designer that put together the pieces. And it's more built bottom-up, with cells that interact. And here you see this is a movie from Philip Keller's lab at Janelia from the post-implantation mouse and the development. These cells are interacting. There's never a single cell that takes a decision. It's a single cell in between a population of cells. And so a lot of our question is like, how can this happen? And we know that a lot of it is really genetically wired. So DNA is instructing the timing, and this makes most of the process during development very precise and reproducible. However, if you consider regeneration and homeostasis in an adult, also different other aspect, has to be self-organized, and we need a very strong space-time coordination of single cells in order to make it robust and contextual. So cells need to have sensing mechanisms to understand their environment to take correct decisions. And so this is what we are really very much interested in the lab, how this space-time coordination happens at the single cell level. So that means that cells have this cell-intrinsic mechanism to sense. These are molecular mechanisms, and every cell is different. We all start from genetically identical cell, but very fast in a population of cells, there is an emergent cell-to-cell variability that really emerge at the population level because all the cells start to be different. You have cells in different cell cycle stages. There are different transcriptional state. And so every cell is different, and every cell senses its environment differently. And this creates different probability for some cells to differentiate. Some then steps, like in this case, I call it symmetry breaking. Some cells differentiate, start changing behavior, and this behavior really drives the emergence of tissue-level properties. However, this is really a problem about scales and is really inherently complex. What I mean about scale is that you might have a very fast mechanism, like a receptor at the plasma membrane, a signaling cascade, that happens very fast here, that has consequence many days after. So at the timing, there's like a very big difference in scales. There's also a difference in spatial scale. There are things that happened inside the nucleus, bursts in transcription, that might have effect very later on in very long, different scales. So we need crossing scale technologies both at space, time, but also functionality. We need to have a system where we can follow every single cell. We can perturb every single cell to understand the molecular mechanism, really to get at the mechanism. And so all this crossing scale technology. And I think organoid has been said already this morning, are a perfect model to have this crossing scale technology at the functional level, where we can take stem cells and have them grow. For example, here you can take an intestinal stem cell from a mouse or a patient from a biopsy. And in more or less 10 days, you can recreate a fully formed intestinal organoid that has a lot of the functionality of the real intestine, so has the same spatial organization and the same functionality. However, it's in vitro, and we can have screens with it, and we can really modify it. Moreover, we can really image it, so look at single cells, how they grow, and how they develop. What is still unclear, and even if this is one of the first organoid that was developed, and this was done in 2009, was still not really clear how this grows and how does a single cell can grow it. And then I will come at it at the end. I think for all this system, we still need to understand how normal is and how we can grow organoids. And again, this is a problem about scale. And being in really complex, we need both multivariate and multiomics type of approaches to understand it. For example, and going back to the protein, yes, RNA is not the only one, there's also protein, and their localization. And so there are different methods that have been developed, both iterative method, and I won't go in detail, but they're both that can look at single cells in their spatial context and look at multiple RNA and protein composition. I will just go a bit in detail in one. So here you see it's a population of cells. For every cell, you can image 40-plex protein localization and abundance. For all this, you can have at the pixel level a feature vector that then describe the protein localization, abundance. And these pixels can be clustered and remapped at the full scale. So for a very big piece of tissue or for a full big organoid, we can have 40-plex or 50-plex quantification of protein abundance. This was recently used to characterize the formation of these intestinal organoids, as I showed. And these data can now be used to infer trajectory similar to the trajectory that are inferred from single-cell RNA-seq, but for imaging data. So we can start from imaging data where we have not only information about the abundance of the protein, but also their localization and what is the neighboring effect of every cell to create trajectory of organoid development. However, this was very interesting because we could see that organoid formation, and this I will go very fast, doesn't have stem cells in the beginning of their development. And as you can see here in green, these are the stem cells, and intermediate time point, there's no stem cell. And we thought that was very surprising, so we start from a stem cell, we go to a fully formed organoid with a stem cell niche. However, in between, we go through a stage where there are no stem cells. And what was very important at this point, going back to the multiomics, is to be able to really integrate data set. So we had a very strong spatial and protein-based analysis, but we needed to know more what happens during these first days. And there we really combined single-cell RNA-seq data set, where we could then figure out that this intermediate stage of organoid development is a regeneration process. So organoid development from a single cell is not driven by a developmental and stem cell mechanism, but by a very transient regenerative process. And this is what we see. And I just want to show this slide for stress one point. This very transient step of organoid formation depends on YAP activation. And this is happening in a very short period of time, exactly between 46 and 50 hours of organoid development. So these are fast events that we cannot capture with single-cell RNA-seq in the way we are doing it right now. And we really need to combine both the single-cell RNA-seq with dynamic analysis of spatial method to really pinpoint this mechanism. But again, these trajectories are still an average. We don't know what is exactly happening to a single cell why it's going through this trajectory. We don't know what are the different processes that can affect this decision-making and how different processes like the cell cycle or cell-cell interaction can determine some of the decision and changing the probability for some cells to go through one step or the other. And for this, I think we really need to go to a more dynamical, in total imaging of what are our system, and for this organoid are perfect. And light sheet has been a very strong changes in the way we do our imaging because it's very fast, for low toxicity. And moreover, what is happening is that now we can really grow organoids in a very confined environment. For a very long period of time, we can do 2 or 3 weeks of imaging, of continuous imaging. And here you can see again this organoid growing from a single cell. Here is a Z-plane, and this is a full projection. And you can see that the cells divide, and we can follow the dynamics of the different process. We have a very synchronized division. The cells divide very synchronously between 4 and 8 and 16. And between the 16 and the 32 cell stage, there's the first changes where the first cell differentiate and start recreating the organoid. And you will see here the formation of simultaneously 3 crypts during the organoid formation. Clearly, this is a very nice movie to look at, but how can we then relate this to all the other data sets? So we really need to be able to track the single cells, and now with machine learning and different computer vision method for the analysis, we can then start tracking every cell and go back in time to follow the development of this organoid. And you can see here, you can really follow the final cell and how it developed during the organoid. And so what I think is that we are now getting ready to understand both for especially in organoid, to have predictive model of single cell, but really single cell, during crucial decision-making moment, and understand how these cells move through a trajectory and how these decisions are influenced by different factors, and understanding the molecular mechanism underlying this decision-making moment. I think that what we have now is really this bridging scale technologies, both for biological, so functionally, the organoid, and technologically to have imaging methods and single-cell RNA-seq method that can really scale over the different, that can be bridged over the different scales. We have in vitro models that are getting really always better in regeneration and disease. And we are at the moment where we can look also at protein level that makes us much closer to phenotype and not only to genotype, especially in this very transient state. And there's still, I think, a lot of challenges. As I think one, and our in vitro analysis of the intestinal organoid was quite clear, is really to understand this in vitro model system. We have been using intestinal organoids for a while, and now it's, I think, quite clear it's more a regenerative system. So I think that for all this organoid, we need to understand better how they're formed and what they are really recapitulating. Having multivariate and multiomics at the type of technology at different scale, but also understand where are the crucial points where we can integrate the data set, and especially the dynamical aspect. And with this, I would like to finish, thank my lab, and thank you all for your attention.
Thank you very much, Prisca. Questions?
So this was a great talk and also a great paper that you recently published in Nature. So at the same time, I found it a little bit worrying because most of us here in this room assume that organoids are a good model for tissue in homeostasis. So what your paper shows, no, the intestinal organoids are exactly not this, but this is a regenerative response. So basically this means that for every kind of organoid we now have to say, show what kind of process it mimics in the body, right?
So let's say the formation of organoid mimics regenerative. However, a big form organoid really recapitulate more homeostasis, and this is quite clear. So it just gives us an opportunity that we can now use the organoid in two different ways. If we want to look at homeostasis, we looked at a fully formed organoid, let's say a day 10 organoid. However, if we want to look at more dynamical process like the regeneration and the YAP-dependent regeneration of the intestine, we look more at the initial days. So yes, in part, we need to characterize better our system, but I still believe that fully formed organoid have a full recapitulation of the homeostasis situation. So a big form organoid is an homeostatic model.
I actually have one, Prisca, because I mean, as we've seen, it's very important to learn how to track cells over time. So, what is your comment? What can you tell us about, developing strategies to follow multiple markers in live cells in organoids? I mean, I think that's going to be the next frontier. So, I don't know if you can say something.
Yeah, so I think that multiplexing live will be one of, I think, for example, having different reporters targeted in different compartments could be an interesting approach. So having some reporter that are targeted to the nucleus, even if they have the same fluorescent color, that could be one. The other one is really try to track the different cells, but clearly the multiplexing capabilities at that point will probably stay in one hand, at least for a while.
One more question, Nicholas.
Yeah, so I really enjoyed your talk, actually.
Thanks.
But you must have lots of, I would imagine, you're also looking at diseases in your lab and growing organoids from them. And so you didn't really talk about this, but can you maybe share with us what kind of phenomena you see or you are interested in?
Yeah, so we look at the different model system for inflammation, for example, and so we have some, but I think some rare genetic disease, some cancer models also, some colon cancer models, and it's still, I think, a bit on the early side for the disease model on the organoid to fully understand what's happening. So we are trying to really use similar approaches on the cancer, for example, on colon cancer. We see very similar mechanism of YAP and this variability in YAP, especially in early moments. We are trying also to combine and having co-culture, having an immune system. We have a co-culture of cells, but we are still at the beginning. We don't have